Well, hello everyone, I am Suleman and I am here to teach you about the cell cycle, or we can say cell division also. So starting with the topic, the very first and common question is why actually cell division occurs. I am going to tell you or explain you about this by giving an example. Let's suppose you are living in a very good city like Lucknow, and the whole city is actually under control of district magistrate. For example, 10,000 people are living in Lucknow and one district magistrate is handling each and every project of the city. And for some reasons, there are people who are getting migrated into your city, and now population is getting increased. Then what will happen? The burden on district magistrate will going to increase, and that will leads to that district magistrate to part the city into two parts. That means he has to divide his work in between two people so that he could do the work more efficiently. Likewise, in the cell, whenever cell continue to grow, because it is a simple characteristic of every living organism that growth and reproduction is actually a very common feature of living organism, that leads to the cell content increase. And if a cell content is getting increased, then that will lead to the increase for the burden of nucleus. And nucleus is because the brain of the cell which governs each and every aspect, each and every work of the cell. So the burden on nucleus will get increased, and that will lead to the cell division. Now nucleus will try to divide its work for in between two, and for that reason cell division will occur. Now we are going to discuss why actually cell division occurs. This is the main basic example which I have just told you, and I hope the picture is very clear to you. Now I am going to tell you more scientifically. Let's suppose we are going to talk about cell division. We know if there is a cell, then there might be the presence of nucleus. Why there might be? There must be the presence of nucleus, and there is cytoplasm. This is C is cytoplasm and N is nucleus. Now if nucleus to cytoplasmic ratio, if it is getting increased, then it will lead to the division of cell into two daughter cells, and these two daughter cells formed because of cell division. Now cell division is actually a part which is occur by a process, that is what we call cell cycle. If we talk about the cell cycle, then it is the main event in which a cell duplicates its genome. Genome is cellular content or cellular data which is getting incorporated inside the thread-like structures. In nucleus, that's what we call chromosomes, and chromosomes have all the data by which a cell is actually having its function, like secretion of enzymes, hormones, or the cell if it is a part of any specific organ like liver or kidney, then every cell has a specific function, and that function or that secretions which are getting governed are actually incorporated inside its genome. So I was talking about the cell cycle. It is the event by cell duplicate its genome, synthesize its main constituents, and ultimately leads to the division and form two daughter cells, and that's what we call cell cycle. And these two daughter cells are actually getting formed because of cell cycle, and this cell cycle is actually a coordinated event. What is coordinated event? Coordinated event is something in which each and every step is actually getting incorporated with one another. Every step is actually under control and will only be done after the one process or one step. So for cell cycle we have some specific points which are mainly given in your NCERT textbook. I am going to quote them. The very first common thing is cell growth is a continuous feature, so it doesn't matter what phase we are talking about. The cell cycle is actually governed by specific proteins, that's what we call cyclin proteins, and there are specific enzymes, that's what we call CDKs, cyclin dependent kinases, which actually govern the cell cycle. There are some specific examples which are given in NCERT, that's mammalian cell is actually take time 24 hours approximately to divide itself, and yeast cell require only 90 minutes, as well as E. coli which requires only 20 minutes. Now we are going to talk about the different phases of cell cycle. So in cell cycle I am going to teach you in a very specific flow chart manner. So cell cycle, we are talking about phases. So phases of cell cycle are mainly two types, or we can say cell cycle is comprises of mainly two phases. The first important phase is interphase, and the second important phase is M phase. Interphase, somehow we call it is the resting phase, but it is not actually resting. Let's suppose if a cell is going to divide, then it will always prepare itself for division. Okay, so in interphase we are going to discuss about sub phases of interphase, which are actually two gap phase and one S phase. Two gap phase are G1 phase as well as G2 phase, and in between both there is S phase, and S stands for synthesis phase. And in M phase there are specific parts or sub parts of M phase in which cell will going to divide itself. In interphase cell is preparing itself for division, but in M phase cell will going to divide itself. Okay, so first of all we are going to talk about interphase. It is it takes 95% of the cell cycle time, and rest of the time, that is 5%, is actually taken by the M phase. M stands for mitosis or mitotic phase, we can also say it mitotic phase. M stands for mitotic. So it takes 95% and it takes 5%. Interphase is actually divided into three parts, that is G1 gap 1 phase, S phase, and G2. If I should draw this cell cycle, then you would understand it better in a better manner. So I am going to draw a cell cycle. Just because it is a cycle, so we are going to study in a cyclic manner. Let's suppose this circle represents cell cycle, and I am going to divide the parts of cell cycle like the quarter part of this circle like this is actually having one phase, and we have to divide this like this. The first phase is G1 phase, then it's S phase, and not like this but requiring very lesser amount like this. This is G2 phase and the rest is M phase. Now you all must be wondering about why I have made two concentric circles. So actually this part is representing the interphase, which is actually having the 95% of the time of whole cell cycle, like this. Okay. Now this M phase is actually divided into its subparts, that is, I am going to represent in a abbreviated manner, that is P, M, A and T. PMAT. P stands for prophase, M stands for metaphase, A stands for anaphase, and T stands for telophase. So these are the four subparts of M phase. Now we can see this whole cell cycle is actually divided into two main phases. The first phase is G1 phase, S phase and G2 phase, which together comprises interphase, and the next phase is M phase, that is mitotic phase, which also have its subphases, that is prophase, metaphase, anaphase and telophase. So as we have discussed about that, interphase is actually the resting phase. It's not actually resting, but cell is actually getting prepared itself to get divided, and for preparation cell is metabolically active. So now we are going to discuss about the G1 phase. Okay, so if we talk about the G1 phase, then what happens in G1? G1 stands for, first of all, gap 1. G2 stands for gap 2, and S stands for synthesis phase. Okay. In gap 1 phase, we are going to discuss that if a cell is getting divided in M phase, then it must have the more amount of protein, the more amount of ATP, the more amount of RNA. So there might be increase in ATP as well as increase in RNA, all types of RNA like mRNA, tRNA, rRNA. Why? Because all these RNA helps to decode the information which are present in the chromosomes, and ultimately it will decode and leads to the specific content, or how cell will going to divide, or how cell will going to form its new component, or whatever cell will going to perform. The ATP is getting increased, RNA is getting increased, protein content will going to increase, because if a cell is getting formed, then we all know that cell membrane is made up of protein, so there might be need of protein, and for that protein content must be increased. Now we are going to discuss about S phase. So S phase is actually synthesis phase, and in this phase mainly DNA replication occurs. And what is DNA replication? The term replica means copy. If DNA is getting formed within the cell, that means it is getting actually copied within the cell, so that whenever cell will going to divide, then there might be an original copy and there might be a duplicate copy. If both the copies are present, then the copy which is duplicate copy will going to form the new cell and the original with intact within the cell. Okay. So S phase is actually for the synthesis of DNA, and that synthesis of DNA is actually the replication of DNA. Now, if we want to understand how actually replication will look like, or how actually cell content will have its copy, then you all have studied about the structure of chromosomes in class 9th. So if we see, there are three terms. One is chromatin, one is chromosome, and one is chromatid. We should understand, if a cell is there, a nucleus is there, and the thread-like structure is present inside the nucleus, then that thread-like structure is actually chromatin. When cell will start to divide, or cell will start to enter into cell cycle, then it will leads to the condensation of these chromatin threads, and that condensation will lead to the formation of chromosomes, and these chromosomes have this structure. This structure is mainly, in some books you could see like this only, and in some books you could see like these. So we could understand this central region is actually the centromere, and these are the two sides of chromosomes. This first construction is known as centromere. If that's what I was saying, in synthesis phase replication of DNA occurs, and we all know that DNA is present inside the chromosomes, and these chromosomes are actually made up of DNA as well as histone proteins. And I have just talked about in gap 1 there is increase in protein, that means increase in protein, that protein might be the histone protein also. Okay. If a cell content is getting increased, then the content inside the chromosome will also going to increase, and whenever cell chromosomal content is getting increased, that means DNA plus histone both are getting increased, and if both are getting increased, these bands, or we can say these arms of chromosome will bulge and leads to the formation of structure like this. Okay, and now we have one this side and one this side. We could easily understood the one is original copy and the other one is duplicate copy, which is actually getting doubled in gap 1 phase. in replication or synthesis phase. Now in synthesis phase, replication occurs, and that leads to the increase in the content of chromosomes, or we can say the replication of DNA or copying of DNA occurs. Now in G2 phase, we are going to discuss that again. Gap 2 is also for the increase in protein content. If we can say the cell is getting divided, then new cell membrane will also going to form and leads to the increase in protein content, as well as there are specific ATP consumption also takes place. How? If I am going to draw the structure, how actually the cell division occurs during M phase of cell cycle, then you will get a clear picture why the number of ATP is required in more amount in gap 1 and gap 2 phase. So I hope you all have understood about the cell cycle, different phases of cell cycle. Now I am going to tell you about the M phase in a different manner. Okay. Now I am going to erase all the portion and going to tell you about the M phase. One more thing I have noted down here, the chromatin, chromatid and chromosome. I have told you about the chromosome as well as chromatin. The chromatid is actually the structure of chromosome. These two different arms are known as chromatid. This whole thing is chromosome, and that thread-like structure present in the nucleus is chromatin. The important thing is chromosome will always occur whenever cell will going to divide, because chromatin will going to condense and form chromosomes. For example, there are 46 chromosomes present in a mammalian cell or in human. So these 46 numbers will going to appear when cell will going to divide. The chromatin will get condensed and forms 46 chromosomes. Now we are going to discuss about the M phase or mitotic phase. The M phase is actually having its 4 phases. I have just told you, that is PMAT in an abbreviated manner, PMAT, that is prophase, metaphase, anaphase and telophase. You could understand this topic by having the diagram how actually the M phase occurs. So the important thing is, if I am going to draw the structure of a cell having nucleus, then this is the chromatin, and these chromatin will going to condense if it will going to enter the cell cycle, and it will form chromosomes. And in the cytoplasm, we know that there is a specific cell organelle that leads to the cell division process, that is centrosome. And these centrosome will going to divide into centriole and move to the every 2 poles and leads to the formation of specific spindle fiber which are also made up of tubulin protein. And in G1 phase we have studied that cell division requires a specific gap phase in which cell starts to prepare itself, and that tubulin protein will going to form in that G1 phase also. Now this is the normal cell, it will undergoing the cell cycle or cell division, then this chromatid will going to start to condense and form chromosomes. So I am going to draw the structure. Now in prophase we could easily understood this nuclear envelope will going to rupture like this and leads to disrupting of nuclear membrane and increase in cell chromosomes. So, and as well as these centrosome will start to move to each and every pole in the form of centriole like this, and they will start to form spindle fibers, and there is presence of chromosomes like this. Okay, let's suppose I am taking 2n is equals to 4 for this example. So there are 4 chromosomes, or diploid condition of this cell is having 4 chromosomes. Then this is the prophase in which how many things occurs? Nuclear membrane disrupts, chromosomes condensed, centrioles moves to opposite poles. Now in metaphase these chromosomes will going to align on equatorial condition. Acha, one more thing, we are discussing about the animal cell in which these centrosomes are there, but in case of plant cell there is no centrosome, there is a specific condition occurs, that is cell groove formation, and that leads to the formation of cell plate which helps in the cell division in case of plant cells. But in case of animal cells, centrosome is there which is getting divided and form centrioles, and these centrioles align at each pole and leads to the formation of spindle fibers. These are the spindle fibers, and these spindle fibers actually connect the chromosome, both part of the chromosome, the chromatids of chromosome, and starts to align them and pulling them towards each pole. Now in case of metaphase stage, these chromosome will going to align at the equatorial plate. Let's suppose like this. The chromosomes are aligned on a specific manner in the middle of the cell. That stage is actually depicting the metaphase stage. The chromosomes aligns at equatorial plate. Now in case of anaphase, these chromosomes which are actually having this structure like this, now in anaphase these will going to divide and half of the chromosome will split and will go to one end or one pole, and the other half will going to the another pole. The other half is the original part, and this half is the duplicate part, which is actually formed in the S phase during the replication of DNA. Now in anaphase, same diagram will occur like this. And in this case, the chromosome will going to split like this, and this, they will go to the each and every pole opposite to one another like this. They will move towards each and every pole. So in case of anaphase, the chromosomes split and moves to the opposite poles. Now at the end, that is telophase, in this subphase again we are going to discuss that this part in a cell, again nuclear envelope will start to form. Now the every strand or half of the chromosome which are getting splitted and get moved to the each and every pole will going to form a separate identity like this. One, two, three, four. 1, 2, 3, 4. As we can see, this nuclear envelope is actually getting reformed in telophase stage, and chromosomes are getting parted on the both side, and now this division will occur, division of cytoplasm will occur, and the cell organelles will also going to divide, and this leads to the further formation of two new daughter cells, and each is having the at its own cytoplasm as well as nucleus. Same in the other cell, that is nucleus and that is cytoplasm. This is the part of cell cycle which I have just told you about. And now if we are going to discuss one more point, you must have read about the G0 phase also, that is G0 or G0 phase. What is this? Let's suppose if a cell has completed its cycle and completed G1 phase, S phase, G2 phase as well as M phase. Now that cell will going to have its own function, like now two daughter cells are getting formed, and these two daughter cells will going to perform or get incorporated to form a different types of cells. That's what we call differentiation. If somehow these cells will not going to come back in cell cycle for any reason, they want to get destabilized or they want to go into a resting mode. If they are not metabolically active, then they will going to enter a specific phase, that's what we call G0 phase or G0 phase. The question arises कि G0 phase आता कहाँ पर है? Again we are forming this cell cycle, Let's suppose that was our cell cycle. That was G1, that was S, that was G2, and that was M. At this point, new cells, two new cells are formed. These are called daughter cells, must have equal size. These are the daughter cells. Now these daughter cells will go into a process, that's what we call differentiation. That means these cell will going to form specific types of cells. If these cells will going to metabolically inactive, then they will enter again into G1 phase, but we will represent it as like this. And here we will write G0 phase. Why we are making this arrow from this area? Let's suppose if we form this line from this point, then it will represent like, it will make a confusion among the students that it might be chance that these cells will again entering into G1 phase and now they are exiting G1 phase, and might be they will come back again in G1 phase. No, this will not occur. The main thing is, now the differentiation process occurs. If this cell is again will want to divide again, then it will get incorporated into cell cycle again. That's de-differentiation. And for de-differentiation, cell will have to enter the cell cycle again. But I am saying these two daughter cells will metabolically inactive, then they will not going to enter the cell cycle. That means they will exit the cell cycle from the G1 phase, because they have completed G1, S, G2, M and formed two cells. And these cells will going to exit the cell cycle and go to a resting phase. That's what we call cell is metabolically inactive. That's what we call G0 phase, or it is also known as quiescent phase. I hope you all have studied this topic very well and understood what I have told you. If you actually have understood this topic very well, then please like, share and subscribe. Thank you for watching.